Start recording. All right, we are going to conclude the nervous system. We're going to pick up where we left off. And again, some of these things I'm going to go through a little more quickly. I'm going to pull out some of the highlights, some of the things you really want to know about. If you've been watching the review videos, then you have probably heard some of this stuff already, which is good, but we're going to hear it officially now. So if everyone is ready, I'm going to get started now. So you should be able to see my screen right about now. Oops. And this is kind of where we left off here. Yeah. The nerve impulse. This is that electrical signal that is traveling down the neuron. And remember, electrical signals are just a difference of positive and negative charges. So this is happening along the membrane. It's not actually going like in the center of the axon, zipping down. It is traveling along the membrane, so it's sort of on the outer part of the cell. And it is following a wave-like pattern in that it's moving in one direction. I think we may have talked about that a little bit already. The resting membrane potential is basically the cell at rest and the membrane at rest where nothing is happening. Then we're going to see a change with the amount of sodium ions coming in, flexing in. That is going to change the inside of the cell, just on the inside of the membrane, making it a little more positive. And that's going to continue happening in one direction. And here you can see these different ion channels. And unfortunately, they've chosen colors that were relatively close together with purple and pink. You can see how there's a lot of sodium on the outside of the cell exterior. This is the outside, and this is the inside. And the cell membrane with the bilipid membrane, you can see these channels that go all the way through. And that sodium is going to want to kind of go onto the inside because there's a lot more sodium on the outside than there is on the inside. And if you remember, we talked about diffusion of particles wanting to go from where they are many to where they are few. So there's kind of this drive, the sodium kind of wants to go in that direction. But it has to go through these channels, which you can see the ones in the middle, the green ones, the closed sodium channels. Those are going to have to open up to allow sodium to come in. So something's going to have to trigger those to allow them to open up. So if you look at this potassium channel, for instance, you can see how it's closed at the outside, and this one is open, which is allowing potassium to move from the inside to the outside. Again, it's going with its concentration gradient. Okay, so let me go right to the action potential. That's what's being demonstrated here. And I know it's difficult to imagine. So first think of that blue tube as being like the axon of the neuron. And you can see there's a lot of sodiums on the outside, not as much on the inside, a lot of potassiums on the inside. So what's going to happen is sodium is going to move into the cell through those channels. Something's going to cause those channels to open up, and sodium is going to move inside. That's going to make the inside of the cell more positive, and especially right along the membrane. So just right on the inside of the membrane. There's going to be a hesitation, and then potassium is going to move out. And they're demonstrating this here. So the resting membrane potential at minus 70 millivolts. And you can see it. You can see right here, minus 70. Then there's something that starts to change the membrane. Sodium starts to come in, and you notice that area right here where it just starts to increase a little bit. In order for this big change, and you can see it goes up above 30 millivolts. In order for that to happen, it has to get past that dotted line. So you see it's going from minus 70, it's becoming more positive. If it suddenly stopped at minus 80, then it wouldn't be enough for this whole rest of the electrical charge to occur. And there would be no change. It would be as if nothing happened. So there has to be enough positive charges coming in to break this and get it right to that dotted line. And then once that happens, well, then the channels will really open up and sodium comes flooding in, which is why we see that spike in electrical current there. And that dotted line is called the threshold potential. This is sort of the part that it has to get to. It has to get to at least that charge, and then suddenly everything opens up, sodium comes rushing in. Now we have a big change. Now we have an action potential. We have the actual electrical charge happening. And what's breaking down here is what is going on at each of these points. And once it hits that threshold, that's what causes those voltage gated sodium channels to open up. And now the sodium comes rushing in, and we have a big change in the electrical current. The action potential is an all or none response. In other words, it has to meet that threshold. It has to get to that point. And you can see here in the magnitude of the local depolarization exceeds a limit called the threshold potential, about minus 59 millivolts. That's when all these other sodium channels open up and sodium comes just rushing in. And so you have all these positive charges, which causes that spike. Then eventually we have to go back to where we were before, back to that resting potential. So we go through this process of what's called repolarization, getting things set back to the way they were. And you can see that happening here. Then there's something called the absolute refractory period, or just the refractory period. This is a phrase that gets used a lot, the refractory period. But when we're talking about the action potential and the current running along neurons, this is important, because in order for the action potential, that electrical signal, to continue going in one direction, the membrane has to have a moment where it cannot be depolarized. It can't suddenly depolarize again. So we want the current going in one direction. Notice the red arrow on all these. And again, we're going to just think of that tube as the axon of the neuron. That red arrow is the electrical message. That's the action potential. That's the electrical message that's getting sent down the nerve. And all it is a change of the positives going inside, becoming more positive on the inside of the membrane. Now, you see here, it becomes more positive right here. And then what we want to happen is we want this area to then become more positive, and then this area, and then this area. What we don't want to happen is we don't want to suddenly have a shift and for these positives here to suddenly go back inside again. or as you can see in this one. So you can see where these positive charges are now reset back to the outside of the membrane. What is stopping them from going back in here rather than these positives going in here? There is a slight period of time where the membrane resists that. It doesn't allow for it, just for the briefest of moment. However, that briefest of moment is enough to cause these positive charges to move in and not allow these to move in. So that means the signal is going to continue following that red arrow, going in that direction. That's the refractory period. And that's an important component in maintaining this unidirectional signal. Remember, this is just an electrical signal that's going to go somewhere to tell something to do something. And it's just nothing more than positive charges moving into the membrane. But we have to have them move in the correct sequence. We don't want the signal to go backwards. And that refractory period allows for that. It resists these positive charges here from going back inside again, so that the signal doesn't suddenly start going backwards. The refractory period. It's a good term to know, because when you study this more and more and more, if you're going on to your RN degrees, or if you're going to medical school, or if you're going to nurse practitioner school or something like that, you're going to have to rehear all of these things again and have a much, much better understanding of them. And at this point, like I said, most of what you're learning is going to be memorization. It's not until later on where you start having a broader understanding. So at this point, memorizing, remembering that that refractory period is a brief period of time, and really brief, that allows that electrical signal to just continue in one direction. We don't want the action potential to move backwards. All right. Now what this is showing, remember that myelin insulation. We talked about the myelin insulation? I think we did. That myelin insulation that wraps around neurons. That, I call it insulation, looks like insulation. That kind of acts like insulation. The changes in the membrane occur at those gaps in between each of the areas of myelin. And that's nice, because what it does is it allows for this signal to travel very, very rapidly along the axon, but at a very continuous rate. Those little spaces in between are called the nodes of Ranvier. The nodes of Ranvier. And you can kind of think of it as sort of leapfrogging down the neuron. The synapse, where one neuron ends and the next one begins, and there's a little space in between, all of that is called the synapse. So we'll jump to this real fast just for a second here. Sure, there it is. Okay. So we're going to call this one end of a neuron. We're going to call these the dendrites of the next neuron, cell body in the middle. Remember, the action potential is traveling this way. So I'll do blue. So all of this is the synapse. The end of one neuron, the beginning of the next neuron, and the space in between, all collectively we call the synapse. Just the space just this area we call that the synaptic cleft, or sometimes the synaptic gap, or sometimes the synaptic space, or sometimes the synaptic junction, I guess. You know, that'd be the whole thing. Or sometimes just the synapse. So sometimes people will just call the space alone the synapse, which is fine. But technically, synapse, the word, is going to have to include the end of one, the beginning of the next, and the space in between. So for this class, and for all practical purposes, that's the definition of the synapse. For this class, for all practical purposes, this we're going to just call the synaptic cleft. We're going to stick with what the book calls the synaptic cleft. So we're going to stick with this term. Just so you know, there's other terms you might hear for that. Something else. Now, what they're showing here, what they're demonstrating here, is that some of these synapses are going to be completely tight together, and there's not going to be a space in between for the most part. So the electrical charge, the electrical message, just goes directly from one cell to the next. And then the model that I think we've talked about a little bit already is this one here, the typical synapse. When you think of a synapse, there's going to be the end of one neuron, and then there's going to be either another neuron or a muscle cell or something here, and there's going to be that space in between. Now, the thing about this is that the first one just relies on the electrical current going through. So we have an electrical message going directly from one cell into the next cell's an electrical message. Here, we have an electrical message that comes down the neuron, but then that electrical message changes into a chemical message. That's the neurotransmitter. The neurotransmitter is going to move across that synaptic gap, synaptic cleft, and it is going to bind to receptors, which is going to cause sodium to come rushing in, which is going to change that back into an electrical message in the next neuron, as they call it, the postsynaptic cell. This end of the neuron, notice they call it here the synaptic knob. You might also see it as the terminal axon. You might also see it as the terminal bouton, and then sometimes just the end of the neuron. So there's sort of a bunch of different names for that as well. Synaptic cleft. And understand that is really small here, about 1/1,000,000th of an inch in width. Okay, that's about right. So even when you look at something like this under a microscope, it still looks like they're touching. You don't see a space in between there. So it's a space, but it's really, really small. Neurotransmitters are those chemical messages. They're going to sort of take over and move to the next neuron, moving across that space. There's lots of different types of neurotransmitters. As you can see here, excitatory, inhibitory. In other words, some neurotransmitters are going to say, go ahead and do something. Other neurotransmitters are going to say, okay, do not do something. All right, next one. Oh, I know what I want to say. One more thing here about this. When we talk about things happening in the brain, for instance, people's thoughts, feelings, emotions, etc. If there's something about their thoughts, feelings, emotions that we want to change, for instance, maybe they're not very happy. And it's not because the electrical signal might not be coming down here enough, or might not be moving through this neuron enough, might not be enough of that neurotransmitter moving across here. Because remember, just like in the muscle, when that neurotransmitter binds to these receptors, the more it binds to the receptors, the stronger the signal is going to continue. So if it's a weak binding, it's going to cause less sodium. It's going to be a weak signal coming into the next neuron. You'll recall when we talked about the muscle that the signal comes down the neuron as an electrical signal, and then acetylcholine is the neurotransmitter released from that neuron, goes across the space and binds to receptors, and then it depolarizes the membrane, becomes an electrical signal again in the muscle, and then it goes on to the sliding filament. Remember, there was another guy called acetylcholine esterase, or just choline esterase, and his job was to recycle all this extra acetylcholine that was in the space and bring it back up in. Kind of, I drew him like a Pac-Man. Well, we're going to have something in these other spaces as well that's going to do the same thing. It's going to recycle. So when that neurotransmitter comes across this space and is weakly binding to these receptors, that guy is still going to be doing his job recycling, whichever one he happens to be. We'll just call him the recycler. He'll still be recycling and moving those neurotransmitters back up into the presynaptic neurons so they can be used again, created again, and used again. So what we could do is we could take something to block the recycler. So even though there's only a weak signal coming through here, maybe there's only fewer receptors or something, if we block the recycler, that means all of these neurotransmitters are going to stay in this space longer. And the longer they're there, the longer they are there, the more they're going to bind these receptors. So if we block that recycler, we could end up with that neurotransmitter staying in that space longer and really just hitting those receptors, causing that membrane to allowing the membrane to change, causing sodium to come in. The end result is going to be a stronger signal going to that postsynaptic neuron. So we use this, for instance, in patients who have, let's see, with their muscles, we can block acetylcholine esterase, myasthenia gravis. We can block that recycler. In patients who are maybe sad, depressed, and their brain, we can block the recycler to one of their neurotransmitters. We call those selective serotonin reuptake inhibitors. And that blocks that recycler's ability to clean out all those extra neurotransmitters, which means the neurotransmitters stay in that space and they just keep bombarding all these receptors. That's going to cause a more stronger signal into the postsynaptic cell. That could help elevate their mood, make them happier. So when it comes to things that alter the thought process in the brain, whether they're a prescription or not, everything works at that space. Everything works on somehow altering that part where the electrical signal goes to a chemical signal and then back into an electrical signal again. Somewhere in that, with that space, that is what is manipulated. Severe psychic depression occurs when deficits of norepinephrine, dopamine, and serotonin. Serotonin. Yeah. And this is in the textbook. So you can see here is what I was talking about. Selective serotonin reuptake inhibitors. The reuptake means the recycler. We're inhibiting, we're blocking the recycler, so he's not taking up that serotonin. It stays in that space longer and just binds the heck out of those things. Blocking the uptake of dopamine. This is something that happens from cocaine. And anesthetics. You remember I've been talking about how sodium coming in, 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 and that's going to create a signal that's going to move in one direction. Well, imagine if you cut your hand severely and you needed to go to the ER and they said, we're going to have to give you stitches. And before they give you stitches, what do? Well, they numb the area, right? And how do they numb the area? They inject something like lidocaine. And what does lidocaine do? Lidocaine is a sodium channel blocker. So if we inject something that is going to block those sodium channels, then sodium that would be coming in, sodium coming in, sodium coming in, sodium coming in, suddenly sodium can't go in, which means there's not going to be any signal. There's not going to be any signal traveling up that nerve to your spinal cord and to your brain that says, ouch, that hurts, because it doesn't get that chance to begin. It blocks the ability of sodium going in. Okay, oh, that was interesting. It's just me. A lot here. All right, moving into chapter 12. And don't be confused. Don't misunderstand me. I realize how complicated that last chapter was. I realize that's not easy stuff to grasp the first time you hear it. What I want you to grasp little parts of it. I don't expect you to have a full understanding of it. If you do, that's awesome. But I don't expect that, because it is complicated. This is stuff that you have to learn over and over again. You have to learn it, forget it, learn it, forget it, learn it, forget it, learn more about it, see it in different ways before you really start having a good understanding of how it works. And unfortunately, there's not a really easy way to break it down because it is a bit complicated. So don't think that I'm going to have an expectation that everybody here should be able to describe all this right back to me what I just said. I do not. It's not like learning the bones, right? A week after learning the bones, I could probably have somebody stand in front of a classroom and say, okay, tell me the bones on a skeleton, and they could probably go through most of them, because that's simple. This is complicated. So, all right, going into the central nervous system. Central nervous system is the brain and spinal cord. Very easy. Well, easy. Okay. Nothing easy about the brain. brain and spinal cord. So we'll take a look up here first. When we cut open a skull and look inside, the first thing we see is the most obvious part of the brain is called the cerebrum. And that is going to have a grayish tint to it. And it has all of these folds and grooves. The folds are called gyri or gyrus, singular, and the grooves are called sulci or sulcus is singular. So most of the brain, most of what you see is the cerebrum. Behind and underneath, there is an area that kind of looks like cauliflower here in this drawing. That is called the cerebellum, which has a great deal to do with posturing. In the center of the brain, there is an area called the diencephalon. The diencephalon houses components like the thalamus, the hypothalamus, or in the posterior portion, way back here, we'd find the pineal gland or pineal body, which is responsible for the circadian rhythm, the wake-sleep cycle. Thalamus is a giant relay center, and the hypothalamus is a main control center. It sets the guidelines of the body, for lack of a better phrase. Then we see the brainstem. The brainstem is what connects the spinal cord to the brain. From the top down, we'll say, the midbrain, then the pons, then the medulla oblongata. The pons is the part that has that little bulge in the front of it. Pons is a Latin word for bridge, by the way. And then the spinal cord travels down through the vertebrae. There's a hole in each vertebra. Of course, that hole is called the vertebral foramen, because foramen means hole. And it is surrounded all around by bone for protection until it gets down to just about lumbar vertebra number 1, L1. And then it kind of branches off. It looks less like one solid piece and more like a horse's tail, which is why that area is called the cauda equina, ending way down in the coccyx. So that is the central nervous system. Obviously, we have skull and bone, the vertebrae surrounding these things, but we also have these layers of membrane. And the layers of membrane are around the brain. The layers of membrane are around the spinal cord. The outermost and thickest layer is called the dura mater. The layer beneath that is called the arachnoid mater and mater. And there is an area beneath the arachnoid mater that looks like a spider's web, hence the term arachnoid matter, arachnid for spider. And you can see it kind of looks like a spider web. That's the area where cerebrospinal fluid circulates. And then the pia mater, is the thinnest membrane and is sort of shrink wrapped around the brain. So altogether, these layers make up what we call the meninges. And you may have never heard of the term meninges before, but you've probably heard of meningitis, which is an inflammation of the meninges. So the brain is covered in these. The spinal cord is covered in these.
Dr. Sturgeon, can you allow me to report, please?
Who is speaking to me?
Your favorite.
Wait, chocolate brownies? Those are my favorite. Well, okay. Chocolate chip cookies are probably my favorite. All right, let me see here. Where are you? Oh, you left and came back in, didn't you? There you go. Okay.
Yes, I did.
Anyone else? Let me go through real fast and make sure everybody has the opportunity. I am recording also, by the way, and I'll put this on YouTube. And Hadia, I have no favorites. I like everyone equally.
I don't believe that, Dr. Sturgeon.
Unless you bring me chocolate chip cookies, then, or donuts, or cake.
That's a good note. I'll bring you some chocolate chip cookies.
Yeah, or cake. I'm just saying, putting that out there. Cake is a good thing. As one of my designs reads, only a sociopath would have cake and not eat it. Who says that? They want to have their cake and eat it too. Of course they do. That's the purpose of cake. Of course you want to eat it. All right. What are we talking about? Oh, brain stuff. We're talking about the meninges and the layers and everything. So let's go back to that. Anyone have any questions? Oh, you know what? Keep an eye on the timer. Anyone have any questions? All right. Let's check this stuff out then, because there's some cool stuff happening with the brain. Here is the spinal cord. So let's first look at this area of it. You can see the bone around it. The bone, that's the vertebra. And you should at least be able to pick out the fact that there's this spiked part. That's the spinous process coming off of the vertebra. So that is the one that's in the back. When you run your hand down the center of someone's back, you can feel these sticking out. So that tells us this is the posterior aspect, and then the body of the vertebra here must be the anterior aspect. And look at how small the spinal cord is. This right here is the spinal cord in comparison to the rest of the vertebra. So if you were to get, if you were to get a single vertebra and look at the foramen, the hole in the middle of it, notice how much of the hole in the middle of it, the hole in the middle of it is filled with other stuff. There's layers of stuff around it. There's a layer of fat in there. There's that adipose tissue in there as well. So the spinal cord itself is not a tight fit because there's all these other things around it. If you were to compare it, you'd find out that it's about the thickness of a pen. Maybe a fairly thick pen, but a pen nonetheless, maybe like a Sharpie. That's not really that big. That's a small piece of tissue that is directing and receiving information, everything throughout your body, basically from your neck down. It's pretty amazing. So they have a nice peel back of all the layers here, the dura mater, the arachnoid mater, the pia mater. I want to point something else out to you while we're here. Back in probably week 1 or week 2, we were talking about directional terms and planes of the body and things. And we said that things that are more towards the front are anterior, things that are more towards the back are posterior. But I also included other terms. I said anterior can also be described as ventral, and posterior can also be described as dorsal. And at the time I said you do need to know that those terms are interchangeable. That anterior and ventral mean the same thing, that posterior and dorsal mean the same thing. And the reason for that is because I know at some point we're going to talk about things like the spinal cord. And you notice how they start talking about these parts as not anterior and posterior, but instead ventral and dorsal. So the terminology changes a bit here. You can also see that looking at the spinal cord, the gray matter is on the inside in the shape of an H, the white matter is on the outside. And we have something else here. We have this layer of fat that is unique to the spinal cord. That is called the epidural space. So when that woman is in labor and delivery, and she decides that she wants to feel none of this pain, but wants to get the baby out, then they can administer an anesthetic agent into the epidural space. And that will cause that dorsal area especially to stop sending the signal to the spinal cord and up to the brain. And that way, childbirth is an absolute delight. No discomfort whatsoever. I know you're tilting your head right now saying, yeah, that's not right. Well, if you talk to some women will tell you that that epidural agent injected into the that anesthetic agent injected into that epidural space was wonderful, worked perfectly. And you notice I call it an anesthetic agent introduced into the epidural space, although I know everyone's going to call it an epidural, so that's what I'll call it from this point forward. That epidural, she will say, was wonderful. It worked perfectly. Somebody else will say that they had to have a second epidural, but then that worked out well. And then somebody else will say it didn't work for me at all. And women are sometimes surprised about these things, about these differences. But they shouldn't be, because every one of us knows that medication doesn't work the same in everyone, especially when it comes to fit. And I'm not talking about like antibiotics. I'm talking about like pain medication. We know this because if I have a headache, I take 2 Advil and that's it. It knocks it right out, does a great job. Somebody else has a headache, they have to take 4 Advil, but that's what works for them. Somebody else has a headache, they say Advil doesn't work at all. I've got to use something completely different. I got to move it up a notch. Doesn't mean that Advil or ibuprofen doesn't work. It just means that it works differently for everyone. Doesn't work, or I should say it doesn't work exactly the same for everyone. So we already know this. You already know this. You already know medicine doesn't work the same for everyone. So it should come as no surprise that when a woman's getting an epidural, meaning, of course, that anesthetic agent introduced into the epidural space, that it might not have the same effect as it did on the last patient. And I hope that makes a little bit of sense. I'm not going to worry too much about the inward extensions. The falx cerebri is just a dural fold attaching at to part of the cranium. But yeah, I'm not going to worry too much about those. I talked about the epidural space. That is specific to the spinal cord. The subarachnoid space, that is important because it is in the subarachnoid space where the cerebrospinal fluid circulates. CSF, cerebrospinal fluid. That is the fluid that distributes nutrients throughout the brain and spinal cord. That is made actually inside of the brain in these empty spaces called ventricles, but that is the fluid that circulates around. That's an important one, important to know about. Okay, I talked about the meninges, I talked about meningitis. CSF, I just talked about that. Notice it is created within the ventricles. The ventricles are empty spaces, and technically there's 4 of them. These are empty spaces in the brain. They're not completely empty. Inside, we find these cells called ependymal cells, these type of neuroglia cells. And ependymal cells are going to create the cerebrospinal fluid that's going to circulate around and just distribute nutrients. Now, if the patient is creating too much cerebrospinal fluid, or if it's not circulating properly, it could build up in these ventricles. And that could actually cause the ventricles to enlarge.
Dr. Sturgeon, so we see those like children with those really big heads.
Yes, exactly. Because if this starts to enlarge, that's going to cause this to get the cerebrum here to get pushed up. And in those babies, remember, those bones haven't all fused yet in their skull. So if that brain is getting pushed out and out and out and out because of this increased fluid pressure, that's going to cause the skull to get pushed out and out and out, which is going to cause the head to be bigger.
Can that only happen in children?
Yes, it can only happen in children that the skull is going to enlarge, because once those bones fuse together at those sutures, there's no more enlarging happening. So anything then that takes place inside of the skull that's going to cause things to grow like this is going to cause soft tissue damage on the brain, which is going to cause death.
And is it a way to like drain that fluid out of those like children's heads?
Yes, they would have to put in a shunt, but there's two reasons why it would happen. Either they are overproducing cerebrospinal fluid, or there's a blockage somewhere that's not allowing the cerebrospinal fluid to drain properly. So the hope would be in those children that there's a blockage, because if there's a blockage, that means they might be able to open it and allow the fluid to drain properly. And if that can be done and corrected, then there won't be a problem after that. However, if it's just a matter that they're producing too much cerebrospinal fluid, then they would have to have a shunt put in that would actually drain some of the fluid out. But yeah,
I actually suffer from that, Dr. Sturgeon. I have pseudotumor cerebri.
Oh, okay.
And I had a shunt installed back in 2010. It's Lanice.
Yeah, okay. What kind of signs and symptoms did you have?
It started off with really bad headaches.
Yeah, of course.
And then when it like got worse, my vision, I started having like spots in front of my eyes. And then I had like kind of looked like a shade had came down over my eyes, and the neurologist couldn't figure it out, surprisingly. I actually was diagnosed by an eye doctor.
Huh, yeah. Eye doctors surprisingly make a lot of neurological diagnoses. Like you'll hear about that with multiple sclerosis as well. But yeah, well, those are pretty broad signs and symptoms.
And of course, I had swelling in my optic nerves.
Yeah. And what procedure did they do?
It started off with just spinal taps just to get rid of the fluid, but when that wasn't keeping up, I had a shunt installed.
What would have happened if you didn't install the shunt, if they didn't drain the fluid?
I would be blind by now.
Yeah. Oh, easily. Yeah. Because it's going to be that continued buildup of pressure is going to put pressure on something, right? And that's all soft tissue. The only solid thing in your head is your skull. So as their pressure builds up and builds up and builds up, it's going to compress soft tissue. Yeah, the optic nerve would have definitely been affected. What else? What did the neurologist think it was?
Classic migraines. They put me on every medication under the sun and nothing helped. It had gotten so bad when I was pregnant with my daughter, the headaches had gotten so bad that I was actually admitted to the hospital and sedated. because I just wasn't functionable at all.
Wow. And during pregnancy, we can expect to see an increase in the amount of blood. I mean, a pregnant woman's going to increase her blood volume by 30, 40% or even more. So if there's more volume of blood, there's going to be more fluid. More fluid is going to increase more pressure. More pressure is going to make those headaches go from bad to worse. That kind of makes sense. What kind of follow-up?
I just see the neurologist now because this has been 15 years now. So I see the neurologist every 6 months. I've been doing pretty good. I see the eye doctor regularly just to get my optic, my pressure checked and my optic nerves and.
Do they check it like every year or twice a year?
Neurologist once a year, eyes twice a year.
Yeah.
Just depends on if I'm having symptoms, if I start getting those spots, because every now and then I still get the little spots. If I start getting them, then I'll go to the eye doctor and, they'll dilate me and check me out.
Do they have a word for the spots?
They do, but I don't remember.
Scotoma? Is that the word they used?
No, something else. Maybe something that starts with a G.
That one I don't know. I'm gonna search for the G. I don't know, because typically we say that when a person has these defects in their field of vision, it's as if something is in the way, but there's nothing actually there. It's just a dark area in the field of vision. Is usually referred to as a scotoma of some sort, or a scintillating scotoma you can see as flashes of light, which often accompany migraine headaches.
I get those too.
Yeah.
Not so much anymore, but in the past I did. It was bad. With a lot of weight loss and keeping the fluids down, I pretty much recovered.
Okay, good. It's nothing to mess around with, obviously.
Not at all.
And what did they describe you said your vision went darker, like a shade was coming down?
Yeah, it was like it was a shade over my eyes, like somebody just pulled down like blinds. I could see like shapes and shadows and stuff, but I couldn't make out anything clearly. And their biggest concern was whether I was going to recover from that. Thank God I did.
Yeah.
But they really weren't sure.
Do they call that amaurosis fugax?
Yes.
Yeah. I think that I think, let me think, it's the closing of the curtains, something like that. It translates into the closing of the curtains or something along those lines. Okay. Well, thanks for the input on that. Then you definitely have an understanding of how the fluid works and what it does and what it can do. In young babies, if that happens, their skull will just continue to enlarge as the brain sort of presses against it. This is one of the reasons why when you bring your child in for those pediatric visits, what's one of the things they measure? Head circumference, right? With a tape measure, because we want to make sure their head's growing at the right size and it's not growing way too fast, because that could indicate something like excess amount of fluid. We call it hydrocephalus. All right, good. Good stuff, thanks. Okay, there's the ventricles. So there's basically 4 of them. CSF flows into the subarachnoid space. That's the space that looks like a spider web. Oh wait, there's a picture of it. There we go. That's a good picture of it. You can see the spider web looking area called the subarachnoid space, and the fluid's going to be distributed throughout that area. You see how it flows around the brain from the ventricles. That's a good image. So it's the same fluid. Let me remind you again. This is the same fluid that flows around the spinal cord also. So if they believe that there might be an infection around the brain, for instance, they could check that fluid. However, drilling through skull to get to it might be a little bit dangerous. So instead, what do? They go in through the back and go right into the subarachnoid space and the areas around the spinal cord and drain the fluid that way. We call that a lumbar puncture, or you may have heard it as a spinal tap, because since we know it's the same fluid that circulates around the brain, if there is an infection that's causing an inflammation, the meningitis, you can check that fluid and see if there's any indication of infection, like bacteria itself or increased white blood cells, for instance. Okay. Spinal cord lies within the spinal cavity, as we saw that. Extends from the foramen magnum. Do you remember the foramen magnum? I hope that you do.
Big hole.
A big hole, yes. The foramen magnum is the big hole found in the occipital bone of the skull. That's where the spinal cord meets the brainstem, which of course then meets the brain. And then if you look down here, 2 bundles of nerve fibers called nerve roots project from each side of the spinal cord, dorsal and ventral. Remember, dorsal, same as posterior, ventral, same as anterior. So these nerve roots are going to carry information into the spinal cord. This is incoming information. This is sensory information. Whereas the ventral nerve root is going to carry information away from the spinal cord. That's going to be motor information. In other words, saying, go do something, move an arm, move a leg. It's its simplest forms. So that comes in handy. That knowledge comes in handy later on, but we'll see here. They tell you anterior fissure of the spinal cord, posterior sulcus, so front, back, which means, let's see here, there we are, dorsal root ganglion, that group of nerves. This is all in blue coming into the spinal cord. This is all sensory information. And then in the pink is information going out that ventral root. That is information that's saying go and do something. Go and, like I said, move an arm, move a leg, something like that. So when you think of the spinal cord, we'll see later on that there are these nerve roots coming off of each side all the way down the spinal cord underneath each vertebra. So as a spinal cord goes through a vertebra, just as it goes through it, on the underside of it, there are these nerve roots coming out from the right and coming out from the left. and sending messages throughout the body and taking information into the body. And you can see the cauda equina. Remember I said the spinal cord comes down as this long cord, and then right around L1, it branches off, looks like a horse's tail. And that's actually what cauda equina means. Oops. Okay. I just mentioned it. You can see the spinal cord as a solid part here. And then you can see it has like, now it looks like a whole bunch of long hairs coming off of it. There's still branches coming off of those, but just a bit different in form. In the gray matter of the spinal cord, there are what are referred to as interneurons. These are very helpful because if you are talking to someone in the kitchen and you go to lean up against the stove and you put your hand down on the stove because you think that it's off, but it had just been on a few minutes ago, so it's still really, really hot. As soon as you put your finger down on that stove, what happens? You pull your hand away, you pull your arm away, you maybe even jump out of the way, and then you look up at your hand and then you look down at the stove. The reason why you move your hand before you even feel the heat is because that signal of hot runs up your nerve, up to your spinal cord, and then your spinal cord takes that information before it relays it up to the brain, interprets it as, hey, this is hot, we should move that area away. So the interneuron kind of makes that decision, sends the information to one of the motor neurons, the ventral neurons, tell you to move your finger, move your hand, move your arm, move that side of your body away. And that's actually usually the way you'll see it happen too. If you watch somebody do this in slow motion, you'll notice first their fingers get pulled away, then their hand, then their whole arm, then even their body like will turn away, maybe even jump away from it. It happens in that order because the signals are going down to the muscles in that order that quickly. And then the signal continues up to the brain and it says, hey, you just touched something hot, which is why the next thing the person does is looks at their hand. And then the thought is, well, what did I just touch? And then they look down at the stove. So if you watch it happen in slow motion, it almost always happens in that order. But that interneuron helps with the reflex arc, I'm sure you've heard of. All right, let's see where we are here. Okay, let's take a break before I get into this. I'm not going to go too deeply into some of these different tracks, again, because I think that is better served at a later time where you have a better understanding or you need a better understanding of some of these. So that would definitely work better. I'm going to pause for just a second here. Recording again now, so I'm recording again. Okay, let me switch us back so you can see my screen, which, well, that should happen right about now. All right, as I said, with the spinal cord, the gray matter is on the interior in an H shape. The white matter is on the outside, and which is opposite of the brain. The brain has the white matter on the inside, gray matter on the outside. So what are these things made up of? What are white matter and gray matter made up of? The gray matter, again, is all of the dendrites and the cell bodies of the neurons. The white matter is white because of all of the myelin wrapped around axons. So all the white matter is just axons with myelin wrapped around it. That's why it looks white. And you'll notice with the spinal cord here, they have a bunch of different areas sort of sectioned out, and they're colored here so you can see them. You'll hear these referred to as tracks. And the way I always think of them is sort of special tunnels that are directing specific nerves to and from the brain, sending the signals back and forth that are specific for different types of movement or different types of sensation or different areas of the body. So I'm just going to show you those, and that's it. And you see here referring to them as ascending and descending tracks. The brain. The brain has lots and lots of neurons, 100 billion, give or take. But if there's a lot of nerve cells, neurons, there's even more, almost 10 times as many, of the supporting cells, the neuroglia cells. So here we see in the orange color, the cerebrum. Again, this makes up the majority of the brain, is the cerebrum. Most of what you see, most obvious to you, is that cerebrum. The internal part is a really important part. I mean, this is where a lot of the control centers take place, is the diencephalon. And you can see they've pointed out where one would find the thalamus, the pineal gland, also known as the pineal body or pineal gland, pineal body, as well as the hypothalamus. Then the brain stem in yellow, green, and a light orange, the midbrain, the pons, and the medulla oblongata. And then finally the cerebellum back there in the posterior aspect and underneath. Kind of looks like cut cauliflower from this angle. Okay, so what do each of these parts do? We'll start very quickly with the medulla oblongata. The medulla oblongata is really important in maintaining homeostasis. It's taking in information and making decisions based on that information. So it's a lot like a thermostat in your room. The thermostat is set to a certain temperature, and the thermostat is going to have some sort of device in there that measures temperature, like a thermometer. And when the temperature in the room goes below a set point, whatever it's decided to be, the temperature will be indicated, and that will send a signal to the furnace and tell the furnace to kick on and bring the room temperature back to where normal would be, where we want it to be. So it doesn't set what is normal, it just regulates what is normal. Somebody has to go over and set the thermostat. That's what the hypothalamus does. It's the one who says, this is what it's supposed to be. This is what normal is supposed to be. The pons, so this is the center of the brainstem. The medulla oblongata is the bottom, if we want to call it that, of the brainstem. The pons is the center area. Pons is a Latin word that means bridge. And it does sort of bridge, obviously, the medulla oblongata with the midbrain, the part above it, but it also bridges with the cerebellum. That's the cauliflower looking part. And so it helps in the regulation of posture. In the midbrain, we find an area called the substantia nigra, which is going to make things like dopamine, neurotransmitter. Notice here also, nuclei in the medulla contain a number of reflex centers, including the vomit center. The vomit center is found in the medulla oblongata. That's the area that says, you know what? Time to puke. Let's look at the cerebellum real fast. And you see the pons right here, that little bulging area right in front of it. and they're in communication. So if you're leaning one way and almost falling over, your body is going to say, well, we don't want to fall over, so we need to correct for that. And this is what one of the main jobs of the cerebellum is, about posturing in three-dimensional space. So if you didn't have this, I don't know what that means. If
1:15.
If the cerebellum wasn't functioning, a person would have a really difficult time walking, because walking requires balance. And walking up steps is extremely difficult. It's very complicated to be able to posture our bodies in such a way that we have to shift our weight to one side while also picking a foot up, while also shifting the weight forward and into that side to put the weight down on it, and then lifting up the rest of our body weight with that foot. It's a really complicated, intricate process of just walking up steps. But walking in and of itself requires balance. So there's a family, I don't remember where they live now, dang it. I can't remember exactly where they live. Might be Turkey. Yeah, that's where they're from. There are members of this family called hand walkers, because their cerebellum isn't functioning. So they can talk and they can, carry on conversations, and they can be normal that way. And if they're leaning up against the wall, they'll be fine. But once they have to walk, they actually walk on their hands and feet, not on their hands and knees, bent over on their hands and feet. Otherwise, they'd fall over. It's an interesting study. If you ever look up a video on Turkish hand walkers, you'll see them. And it wasn't until some researchers who were there, I think it was even like a medical student, who said, well, why don't we give them a walker that they can hold on to and walk? So all these people have been walking on their hands and feet. Nobody thought to give them a walker. Kind of interesting. Okay, so that's the cerebellum coordinating all of that. And we can see how this information is coming up, or in this case, going up the brainstem from the pons over to the cerebellum, up into the rest of the brain. From the thalamus is a big relay center, as I said. The information coming back from the brain, down through the pons again, and down the brainstem. All right. Couple of things to note here in the diencephalon. There is a direct connection with the pituitary gland right here from the hypothalamus along that what's referred to as the infundibulum. And the thalamus is a giant relay center. I sort of liken it to one of those old time telephone operators you may have seen in movies back in the 1930s or 40s, 20s, even 20s, where these operators would set up a wall of plugs, and when somebody would call in, they take a plug out of one hole and then stick it in another hole to connect one caller to another caller. So that's how I sort of think of the thalamus. They're taking this information and they're sending it off to where it needs to go into the brain, and then back down to the brainstem, for instance. The hypothalamus, like I said, that's really the guy who sets the controls of everything, what should be normal. It's also a place that really important when it comes to the endocrine system. There's a lot of originating hormones that come from here. And we'll see that hormones work in this chain reaction kind of a fashion, so that there's got to be some place where they start. And a lot of those hormones start in the hypothalamus. The pineal gland, again, we'll see this one again. It creates the hormone melatonin. Do not confuse that. I'm not going to say that yet. Do not confuse that with melanin. Melanin is the pigment in our skin that blocks UV radiation. melatonin is a substance that helps us go to sleep. So when we are exposed to less sunlight, the pineal gland, also known as the pineal body, will release more melatonin. In other words, when it's darker out, this area is going to release more of this melatonin. It's going to make us tired. When it's lighter out, there's going to be less melatonin, and we're going to be more awake. This is why in the wintertime, where we have shorter days and longer nights, you feel more tired, as compared to the springtime when we have longer days and a lot more sunlight, you feel like you're more awake. Hopefully. It does not, wait, let me back up just a second with melatonin. They do sell this as a sleep aid, but understand that this is not like an Ambien. It's not like taking a Benadryl. It's not something you take and it's going to make you go to sleep. They sell it as a sleep aid to help regulate the circadian rhythm, help regulate the wake-sleep cycle. So you can't just say, well, I want to go to sleep tonight, so I'll take some melatonin. It doesn't work that way. I know people think that it does, but it does not. A Benadryl would work better. And there is a correlation between increased melatonin and something called a seasonal affective disorder, or SAD, where people get depressed during, well, times like this. All right, let's move into the cerebrum. The cerebrum. You can see gyri, sulci, I talked about those. The sulci are the grooves, the gyri are the sort of the sulci are grooves, gyri are the curved parts. Yes, that's it. Fissures. Fissures divide each cerebral hemisphere into 5 lobes. Look at the names of these lobes. We're not going to worry about the insula, but look at the other ones. Frontal lobe, parietal lobe, temporal lobe, occipital lobe. We're not going to worry right now about the insula. That is, you can see they're sort of inside. But look at the lobes of the brain. The frontal lobe, parietal lobe, temporal lobe, occipital lobe. So each lobe of the brain has specific jobs that they do. The frontal lobe is where we are going to find the speech center, and the frontal lobe, especially the prefrontal cortex, the newest part of our brain, it is going to help determine the benefits and consequences of future actions. So let me tell you what that means, because this is what has made us successful as a species, being able to do this. And when I say the benefits or consequences of future actions, I don't just mean, should I go to college or should I rob banks, but also moment-to-moment actions. And the example that I always use, if you're walking down the sidewalk on your way to the friend's house, and you come across an area of sidewalk that has been demolished. In other words, a 3-foot by 3-foot square of concrete is missing. There's just a big hole there. Your options are to step down into the hole and then step back out of it, or to try and jump across the hole, or to walk on one side of the hole, but that would put you closer to the road and therefore closer to traffic, or to walk on the other side of the hole, which would put you on somebody's lawn. They might not want you walking on their lawn, but those are the basic options. Or of course, to turn around and just say, forget it, my friend's not that important. So the amazing thing that happens when you're walking down the sidewalk and you come to this big 3-foot by 3-foot hole in the sidewalk, you pause for less than a moment. And then you just walk around onto the person's grass and you keep going. Now, what happened to all those other options? you might want to make sure your children are muting their mics. So what happened to all those other decisions? Well, the brain thought I could step down into the hole, but I could slip. That's going to get my shoes dirty. I'm going to have to use extra energy to step out of it. That's not the best idea. And the brain thought, well, I could jump over it, but that's going to require extra energy. And what if I don't make the jump? I could slip and fall and hurt myself. And the brain thought, well, what if I step on this side of the hole, but that puts me closer to traffic, which is more dangerous. Or I could step on the other side of the hole and just walk on this person's grass and then be on my way. The brain considered all of those options without you even realizing it. It ran through each scenario and chose the best one. And to you, you just walked down the sidewalk, there's a big hole there, and a second later, you're walking around on the person's grass. But your brain went through all of these different scenarios and thought, which is the best and safest way to go from where I am now to on the other side of that hole? Without you realizing it, in a split second, worked out all these scenarios. So this is what has really helped us become so successful. So when I say the frontal lobe helps to predict the benefits and consequences of future actions, it almost, you could say, predicts the future, because it predicts what's going to happen, what you're going to do, what choice you're going to make. It predicts what happens in front of you. You see how the frontal lobe predicts what happens in front of you. The benefits and consequences of future actions in front of you, the frontal lobe. In the front, frontal lobe. All right. The parietal lobes, these are involved with sensation, like touch, pain, vibration, as well as the gustatory cortex. In other words, when you eat something delicious, you're tasting it with your parietal lobe. I know you think I taste with my tongue. No, you don't. You taste with your brain. Taste is occurring in the parietal lobe. The temporal lobe, this is an area where we find the auditory cortex. This is where sound occurs. But understand, that is different than voice recognition and understanding. So sound, like you hear my voice, in the temporal lobe, it's saying, I recognize that as a person's voice. Maybe even I recognize it as human voice. I recognize Dr. Sturgeon's voice. But then it has to travel to other areas where you actually are understanding the words that are coming from my mouth, where you can actually make sense of my sentences. That occurs in another area. But the temporal lobe is where hearing occurs. Also, the temporal lobe is where we find the olfactory cortex. So that is where you smell. Well, I don't mean you smell like you stink. I mean, like the sense of smell occurs in the temporal lobe. You do not smell with your nose, you smell with your brain. And then finally, the occipital lobe. The occipital lobe contains the visual cortex. This is where you see. You see with your occipital lobe. That is where vision takes place. You don't see with your eyes, you see with your brain. Vision takes place in the occipital lobe. So to remind students of this, I say it's like having eyes in the back of your head, because that reminds you that vision takes place way back here. You don't actually have eyes there, of course, but if you've heard the saying, somebody having eyes in the back of their head, the occipital lobe is where one would find the visual cortex. It is in the occipital lobe where we see. Now, there's a giant fissure that runs down the cerebrum that divides the brain into a right side and a left side. So there has to be some way that those sides communicate. And that is called the corpus callosum. Big band of tissue runs from one side to the other. It says, okay, this is the information I'm getting. I wanted to share it with you. What do you got for me? So now you can see a little more. Oh, here's motor speech, Broca's area. That's one of the, remember I said the frontal lobe, along with predicting the benefits and consequences of future actions. I said the speech center is found in the frontal lobe.
Dr. Stanley,
Yes.
I missed something. You talk about something that runs from one side to the other. What is the name of that?
There's a fissure, a groove
Okay,
that goes from the front to the back.
okay.
And that separates the brain into two sides, a right side and a left side, the cerebrum. And then there's a band of tissue called the corpus callosum, so that the right side of the brain is able to communicate information with the left side of the brain, left side is able to communicate information with the right side of the brain.
Okay, that's the corpus, yeah.
Corpus callosum.
Okay, corpus.
Yep.
All right.
Yeah, so I wasn't sure which part that you missed. So let's see. You can kind of see in this picture a little bit. There's a big long groove that runs from front to back of the cerebrum that divides it that way.
All right, thank you.
Mm-hmm. Okay. All right. I know you're looking at this thinking that is a weird looking guy. This is called a homunculus. 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 Make sure I got that right. Homunculus. And basically, what this is just demonstrating are areas of the brain that are associated with particular areas of the body and how sensitive they are. So the areas that are the most sensitive are drawn really, really large, like the big hand or the big tongue or the big lips. Areas that are less sensitive are drawn smaller. So sometimes you'll see this as actually just a guy, like a animated figure almost, that has a really big head and these really big hands stretching out from their body. But that's all this is demonstrating. At this level, again, I don't think it's necessary to make sure you understand the areas, but when you see this picture, just think of it that way. Think, oh yeah, these areas are much more sensitive to touch. You can feel something much more sensitive in these areas than you can in others. That's why they're made larger. Okay, definitely a term I want you to know, consciousness. I want you to know it because I think most everyone has heard of the word consciousness. But if I were to ask somebody, tell me your definition of the word consciousness, they might not be able to come up with a great definition. They might stumble a little bit. You might say, well, it's like being awake. Well, it's a little bit more than just being awake. There's a little bit more to it. So this is one of the words that I definitely have included in the review. I think you probably saw that if you watched the review. Consciousness, state of awareness of oneself, one's environment, and other things around them. I don't say humans because it also includes, you know, animals, trees, maybe even odors. If you're smelling freshly baked chocolate chip cookies. So all of those things. State of awareness of oneself is certainly the most basic definition of consciousness. So to include the environment and other humans is good as well. But it's a good definition to know because, well, like I said, you've probably heard the term before, but you probably didn't have a good definition or or you might not have been able to come up with a good definition for it. Now you can. We're not going to talk about the reticular activating system. Anesthetic drugs produce an altered state of consciousness sometimes, yes. Sleep, state of seeming unconsciousness. Aphasias. Language defects, as a result of lesions in the speech center. So there's different types of aphasias. In some types of aphasia, the person might have clear thoughts. They can think clearly. I know exactly what I want to say. But when I speak, what comes out is something like, or another type of aphasia might be, I can speak perfectly fine, and as I'm speaking right now, but when people speak to me, what I hear is wah, 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 wah. Sort of like a teacher in a Charlie Brown cartoon, if you've ever seen those. So this one I include because you can see aphasias in patients who've had brain damage of some sort, maybe a cerebrovascular accident, which is what we call a stroke, a CVA, cerebrovascular accident. So you'll see the term aphasia, some type of aphasia, receptive or expressive aphasia, or receptive aphasia, or global aphasia. So you'll see that. And another important one, emotions. Most people are familiar with emotions and their emotions. But here's the thing about the brain and the emotion center. There's not really one part of the brain that is just for emotions. It actually requires several areas of the brain that work together to help us have these feelings. What do they include? Anger, fear, sexual feelings, pleasure, sorrow, etc. So instead of saying, well, this is the one part of the brain, we call it the limbic system. The limbic system. The limbic system is the area that makes us experience different emotions. But again, it's made up of several different areas of the brain. I think that's good. One of the things that's demonstrating here is the nerve tracts that I talked about before, where I said that the spinal cord, you can see these different cutout areas that are, I think of them as like being tunnels where certain nerves are going up to the brain or down from the brain, sending signals up or out. So that's kind of what's being demonstrated here, as showing a cut section of the spinal cord, the midbrain, and the brain. That's all I want you to know about that. I think we are coming close. Okay. When it comes to memory, remember, when it comes to memory, that there's two parts to it. So I made myself laugh there. I said, remember about memory. There's two parts to memory, short-term and long-term. In order for things to go into long-term memory, they typically have to pass through short-term memory. Things don't typically just go right into long-term memory. In order for things to make that move from short-term memory to long-term memory, they have to be important. Your brain has to decide this is important to move it into long-term memory. This is why, one of the reasons why we dream, because that's part of what is happening. Our brain is determining what's important and what not. If it's not important, then it can get rid of it. Short-term memory. Short-term memory is the stuff that happens all day long. And the example that I always like to give, if I drove you down to Wawa and you ran inside to grab a Twix bar, because I'm going to get one of those and you're going to get one, and you're standing in line and there's a guy in front of you who's buying all this stuff and he's paying in pennies and nickels, and he's standing there counting out $200 in pennies and nickels, and you're waiting and waiting and waiting, and you're standing behind them and you look at the back of his coat and it says Tom's Towing Service, or Tom and Jill's Towing Service. Tom and Jill, yeah. Tom and Jill's Towing Service. And you're waiting and waiting behind this guy who obviously works for Tom and Jill's Towing Service. Finally, you get up, you get to pay. You come outside, you get in the car. I say, wow, what took you so long? You say, there's a guy in front of me paying in nickels and pennies. I say, what guy? You go, oh, the guy wearing the jacket with the towing company on it. I say, oh, okay. And then a few weeks go by, and I ask you, hey, remember that you were in Wawa and you took a long time? Oh, there's a guy in front of you. What was on his coat? You would probably say something like, oh, it was like a construction company or a trucking company or something like that. You probably wouldn't remember. You remembered it when you walked outside, but you didn't remember it a few weeks later. Why not? Well, it wasn't important to you. Your brain said, it's not really a big deal. Who cares? So it got rid of it. So how do you get things to go into long-term memory? You have to make them important. And how do you make something important? How do you tell your brain something is important? Repetition. 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 That is how you tell your brain something is important. This is why you hear me say things like, our cells make two things more than anything else. They make energy and they make proteins. And the thing that we need to make energy are glucose, oxygen, and water. And the main sugar that we use is glucose. And you could tell it's a sugar because it ends in an ose. And you hear me say this stuff over and over, and I hope that you say it over and over, because then the stuff is going to move into long-term memory. Just like when you sing the words to your favorite song over and over and over again. And then 5 years can go by and the song can come on the radio and you're singing it again, almost word for word. You remember pretty much every word. The reason for that is because you stuck it into long-term memory because you told your brain it was important. So when you study for these things, you have to tell your brain this material is important. And the way you do that is through repetition. What is that muscle that turns our head towards the center and down just a little bit?
Prayer muscle?
The prayer muscle, yes. What's the name of it?
Diaphragm? That's in the stomach, I think.
There's sternum.
Sternocleidomastoid.
No, okay.
Has two places where it starts, has one place where it inserts. Sternocleidomastoid has two places where it starts, one place where it inserts. Sternocleidomastoid, 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 sternocleidomastoid. And I would stand in front of the mirror and I would point to the sternum, the manubrium, and then I'd point to the clavicle, that's the cleido, and then I'd point to the mastoid process. And as I said, sterno, I'd point there, cleido, I'd point there, mastoid, I'd point to the mastoid process. Sternocleidomastoid, 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 sternocleidomastoid. That's what I would do. And I would do it the next day, and I would do it the next day. And then my brain would say, this is important. we're going to put this somewhere. Now, there are exceptions to this rule in with regard to the repetition part. Many of you aren't really old enough to remember September 11th, 2001. But for those of you who are, like I am, of course, or other people my age, if you ask them where they were when that happened, they will tell you exactly where they were. They might even tell you exactly what they were wearing or what they were eating or who they were with, because that was such an unbelievable event that it created so many strong emotions all at once, like few things would ever make you feel, that the brain just automatically stuck that right into long-term memory, said, holy cow, this must be important. So there are times like that, but there's not many. I know one time I was asked if I remember where I was when Neil Armstrong landed on the moon, July of 1969. I said, I think I was in the third trimester because I wasn't born yet, but I was pretty close. Or people who are older will say they remember exactly where they were when John Kennedy was assassinated. So things that are just exceptional that happen in your life, and not necessarily good exceptional. They could be extremely bad exceptional as well. The brain could say, well, this is important, so I'm going to push it right into long-term memory. Then there's a few people in the world, a small percentage, that remember everything. You can ask them, they can be 50 years old, but you can ask them on this date back in 1974, do you remember where you were, what you were doing? And they'll say, I was in third grade, I was wearing my favorite Christmas sweater, and we were having hot dogs for lunch that day. Now, how could they possibly remember all of that? And it's documented. This stuff is an actual thing. Nobody really knows how they can remember stuff like that. But it shows that the brain does have the potential to do it, which is remarkable. But at the same time, do we really want to remember all that stuff? Are those people remembering all those things at a cost? In other words, is it slowing down other functions?
The bad effects of like having that type of memory.
Sometimes we want to forget stuff.
But I feel like anything that like is traumatic that you would want to forget is something that you're always going to remember anyway.
Well, again, traumatic means exceptional. And like I said, exceptional doesn't mean good or bad. It just means extremely out of the ordinary. So a traumatic event like wartime or an assault, that falls under exceptional event. So yes, that's something that the brain could move right into long-term memory. Not because it's a great thing to remember, but because it is exceptional. So out of the ordinary. So of course, there's some things it's like, yeah, I'd rather not remember that. So when you're wondering how you're going to remember all this information, well, first of all, I have to tell you, my job is to tell you what's important. And my job is to tell you what's important in a way that helps you remember. But I can't do all that work. I can't spend an entire class time repeating sternocleidomastoid, 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 sternocleidomastoid. If I did, you would never forget it. But unfortunately, we also have to learn about the gastrocnemius, and we have to learn about the temporalis and the masseter and the medial pterygoid, all used to close the mouth, and the lateral pterygoid to open the mouth, and the deltoid muscle, and of course, the four muscles of the quadriceps, the rectus femoris, and the three vastus muscles. So there's just too much to cover, but that's what you have to do on your own. I just try to put these other things in here to help you along the way a bit. Like the frontal lobe of the cerebrum helps predict the future, helps predict what's in front of you. The frontal lobe is going to help you predict what's in front of you in the future. It helps predict the benefits and consequences of future actions out in front of you, the frontal lobe. And I try and make connections like this. Or I think I talked about the astrocytes. The astrocytes are the neuroglia cells that separate the blood from the brain. They are cells that have radiating branches. So I said they look like, they remind me of stars. So I think of this group of stars that make up a barrier or a fence that separates blood from the brain. Did we talk about astrocytes? I think that we did. Yeah. Okay. And this is a sensory pathway. The last one we saw was a motor pathway. There's something here. No, okay. All right, hit all the points there. And this would be peripheral nervous system. Okay. The central nervous system, made up of the brain and spinal cord. Central nervous system, made up of the brain and spinal cord. Peripheral nervous system, all of the nerves that branch off of the brain, all of the nerves that branch off of the spinal cord, and all of the branches off of those branches. So everyone here has probably heard the term peripheral vision. Peripheral vision is the vision that's off to the sides, right? You can kind of like see things in the corners of your eyes, the left and the right of you. So central vision is going to be the stuff right in front of you. So central nervous system is the brain and spinal cord. Peripheral nervous system are all those nerves that branch off of the brain and the spinal cord. I'll talk about the, actually now. 31 pairs of spinal nerves, 12 pairs of cranial nerves, and then all the branches off of those branches. Let me show you this very quickly. So I said earlier, remember the spinal cord goes down through the vertebrae, through the vertebral foramina. As the spinal cord descends, there are branches of nerves that come off and go out from the spinal cord underneath each vertebra. So if we look at, well, we're going to start here in the thoracic nerves here. If you look at the vertebrae in the thoracic region, we know how many thoracic vertebrae are there, because it's 12 with a T, thoracic with a T. So if there's 12 thoracic vertebrae, and underneath each one of those, there is going to be a pair of nerves. In other words, one exiting from the right, one exiting from the left, sending signals out to the body, bringing signals back in from the body. If there are 12 thoracic vertebrae, then there's going to be 12 pairs of thoracic nerves. And if there are 5 lumbar vertebrae, there's going to be 5 pairs of lumbar nerves. And even with the sacrum, remember, they're 5 fused together, those bones, but we still number them separately, S1, S2, S3, S4, S5. So if there's 5 sacral bones fused together, there's still going to be 5 pairs of sacral nerves. And we're not going to worry too much about the ends here in the coccyx. So these should all make sense. But I skipped the first one at the top for a reason, because you'll notice something. How many cervical nerves are there? There's 8, but that doesn't make sense because there's only 7 cervical vertebrae. And if I said a pair of nerves extends from underside of each one of these, then there should only be 7 pairs. So why is there 8? Because the first pair of cervical nerves actually comes out above C1, just underneath the skull. And then C2 comes out underneath C1, C3 comes out underneath C2, C4 comes out underneath C5. So there's 1 extra pair in the cervical region. Okay. We talked about the ventral roots and the dorsal roots. The dorsal roots are incoming messages, sensory messages. The ventral roots are outgoing messages, motor messages. Go do something. Some nerves, I think we actually talked about this already. Some nerves have the ability to do both. They can take incoming and they can send outgoing. Oh, that's just convenient, doesn't it? We're not going to talk about the rim, all right? I'm going to talk about a couple of these, at least one of these plexuses, especially like the brachial plexus. But we'll see. In fact, let me show you a picture first. Dude, where's the picture? I don't know. All right, I don't know where the picture is. I know there's a picture here somewhere, but these plexuses have these nerves that sort of come together and then branch back out again. So you'll see that. But there is one thing on here that I do want to include. We'll see this again when we talk about the respiratory system, but I want you to see it now. The phrenic nerve, phrenic, spelled with a PH, renic, the phrenic nerve. This is important because it innervates the diaphragm. Remember, the diaphragm is the dome-shaped muscle that separates the thoracic cavity from the abdominal cavity. And because it is a muscle, it's going to do what muscles do, which is contract, become shorter. Because it is a dome-shaped muscle, when it gets shorter, it's going to become flatter. It's not going to completely flatten out, but it goes from dome-shaped to being less dome-shaped.
Sir?
Yes.
It brings air into our lungs, I believe. Did you say that before?
I'm coming up to that right now. Because when that diaphragm contracts, it lowers the air pressure in our thoracic cavity and therefore in our lungs. And that's what causes air to come rushing into our lungs. So our diaphragm contracting causes air to come rushing into our lungs. And then when the diaphragm relaxes, air passively moves out. It's not being forced out. It's passively just moving back out as the air pressures regulate one another. So because the diaphragm is a muscle, one thing that we've learned is in order for a muscle to do something, it has to be told to do something. So the muscle has to get a signal, this diaphragm muscle has to get a signal that says, okay, it's time for you to contract. And that signal, of course, is going to come from a nerve. And it is the phrenic nerve that innervates the diaphragm. The phrenic nerve innervates the diaphragm. So we'll see that again when we get to the respiratory system. The phrenic nerve. The brachial plexus, deep within the sort of the upper shoulder, like closer to your neck a bit. And those nerves innervate part of the shoulder and the arm. So that's one that you can see get injured as a birth injury when the baby gets stuck in the pelvis, something like shoulder dystocia, and mom's uterus doesn't care that the baby is stuck. So mom's uterus continues to try to push, excuse me, try to push that baby through. In the process of that happening, that area, the brachial plexus, can get stretched. Those nerves can get stretched and damaged. And that baby ends up coming out with a paralyzed arm. It's in a permanent contracture, what we call the waiter's tip contracture. In other words, their arm's straight, their hand is sort of bent up behind them as if they're waiting for somebody to sneak them a tip. Okay, going into some fun stuff here. Oh, man. Fun stuff. Cranial nerves. There are 12 cranial nerves, most of which operate something in the head and neck region. And when we look at cranial nerves, we will notice something. We will notice that they have a name, facial nerve in this case, and they have a Roman numeral. In this case, cranial nerve 7, Roman numeral 7. So sometimes, excuse me, sometimes what you'll see written is a capital C, a capital N, cranial nerve, and then the Roman numeral. Is it important to know cranial nerves? Yes. Is it important that you know them all right now? No. But let me go through them and I'll point out some ones that you should know and you will want to know as the course moves on. Also, some of these nerves are motor nerves. They're just sending signals out. Some are sensory nerves. They're just bringing information in. And some do both. So let's look at the first one, the olfactory areas. The olfactory nerve, cranial nerve 1. And you can see how it's going to pick up information through the crista galli, the perforations in the sphenoid bone, for picking up odor. So this is a sensory nerve, the olfactory nerve. Cranial nerve 2, here we are, is the optic nerve. This is one of the ones that you definitely want to remember. Not that I'm going to test you on it necessarily, maybe on the final exam or maybe on a quiz, but it's just one of the ones that's really important to remember. I'm not going to ever expect anybody to memorize all 12 of them right from the start. But it's much easier if you do it in small parts. The optic nerve, this is a sensory nerve, and it is cranial nerve 2. Now, there's an easy way to remember this. If you look at the Roman numerals, it looks like a capital letter I next to another capital letter I. So it's like two eyes. Optic nerve, cranial nerve 2, like two eyes, like the letter I. So that's an easy way to remember this one. The optic nerve is cranial nerve 2, like two eyes. So it's a good one to remember, and it's an easy one to remember if you remember it that way. The oculomotor nerve is cranial nerve 3. Now, the easy thing about this is it tells you right in its name what it does. It's a motor nerve. Yeah. Cranial nerve 4, the trochlear nerve. This is also a motor neuron, although it doesn't tell you in its name. But you can see that it attaches to muscles surrounding the eye. Trochlear nerve. Cranial nerve 5. This is another really good one to remember, the trigeminal nerve. This nerve does both. It is a sensory nerve, but it is also a motor nerve. Yeah. The trigeminal nerve, you can see, spreads out along the face and the jawline. And it innervates the temporalis muscle, the medial pterygoid, and the masseter, the 3 main muscles involved in closing the mouth. But it also innervates the lateral pterygoid, the main muscle involved in opening the mouth. The significance of this nerve, and this is one of the reasons why I think you should know this one, is because there is a condition associated with it called tic de la Rue. Also known as trigeminal neuralgia, pain along this nerve, sometimes referred to as the suicide disease, because this is a chronic pain that happens in the face and along the jawline. And the pain can be so severe, and like I said, chronic, always happening, the patient just wants to die. So trigeminal nerve, cranial nerve 5. And the way I always remembered this, if you look at Roman numeral 5, it's a V, which is kind of a triangle shape. So trigeminal, triangle, cranial nerve 5. That's how I always remembered the trigeminal nerve was cranial nerve 5. Where's the abducens? Cranial nerve 6, abducens. Some say very funny, but this is also a motor neuron. That's all I have to say about the abducens. Cranial nerve 7, the facial nerve. This is also a good one to know. It does both, first of all. It is both a motor neuron and a sensory neuron. But this one is important because of a condition called Bell's palsy, where part of the person's face will droop, almost look like they had a stroke, but they didn't have a stroke. It's a result of inflammation of this nerve. And it's not really known what causes it. So if we don't know what causes something, we blame it on a virus. And if we don't know which virus, we blame it on the Epstein-Barr virus. But we blame a virus for this condition, Bell's palsy. So we treat it using something for the inflammation like prednisone, a corticosteroid, and then something for a potential virus like acyclovir. So that's why it's good to know this one. You're going to see this. You're going to see Bell's palsy. So So facial nerve, cranial nerve 7, and it does both also. The vestibulocochlear nerve, cranial nerve 8, also known as the auditory nerve or the acoustic nerve, one of those. Or more commonly, vestibulocochlear nerve, cranial nerve 8. You can see it's attached to the labyrinth. Now, it looks like a snail, I know. This is actually called a labyrinth. It is found in the inner ear, divided into 3 parts. The central area is the vestibule. Then we have these areas called the semicircular canals. Those are involved in balance. And then we have the snail-looking part called the cochlea. And that is where we have little filaments that respond to sound waves. And that information is sent along the vestibulocochlear nerve, cranial nerve 8, so that you can interpret, you can hear me, and you can interpret what I'm saying. The glossopharyngeal nerve is cranial nerve 9. Excuse me, and that actually does both. I have to go through my mnemonic to remember that one. But that is both sensory and a motor nerve. It moves the tongue, it assists in moving the tongue, as does the hypoglossal, which we'll see in a little bit. That was 9. Oh, and then here, this one is one of the ones I always recommend people knowing. Cranial nerve 10, the vagus nerve. Vagus is a Latin word. I think it either means wandering or the wanderer. Wanderer or wandering. In other words, it's going on a trip. And you can see that this one goes all the way down into the thoracic and abdominal cavities. That is very different from each of these other nerves. The cranial nerves are mostly head and neck region. This is the one that is the exception and goes all the way down, as you can see, into the abdominal pelvic cavity. The accessory nerves, a motor nerve, you can see here. And then the hypoglossal I talked about already. It's a mostly motor nerve, cranial nerve 12. So, of these, the most important ones to know for right now, and I say right now, but I'm not going to actually test you on them, but they might come up on another test, is the optic nerve, cranial nerve 2. Remember, cranial nerve 2, like 2 capital letter I's, 2 eyes, optic nerve. Let's see, trigeminal nerve, cranial nerve 5, which does both. Facial nerve, cranial nerve 7, which also does both. Vestibulocochlear nerve, cranial nerve 8, which is sensory. And then the vagus nerve, because it's just the oddball, cranial nerve 10. He just does whatever he wants. The cranial nerves are important to know about. Oops, didn't mean to do that, sorry. All right. Afferent is the sensory division of the peripheral nervous system. Efferent is the motor division. So if you think of these electrical signals coming into the spinal cord, for instance, those would be the afferent division. So those signals are arriving at the spinal cord. They're incoming signals, they're arriving. Efferent are the signals going out. They are exiting the spinal cord, for instance, going to tell a muscle to contract. So a good way to remember those is afferent is arriving, efferent is exiting. And it's important that you know those because we will see those terms in other areas of anatomy as well. And afferent is going to mean arriving and efferent is going to mean exiting. So good to know. We've talked about the difference between somatic and autonomic already. Remember, somatic is under voluntary control. Autonomic does things automatically. Like what are they showing here? Cardiac muscle, gastrointestinal muscle, or secretions from glands. Some of these reflexes, there's two that you absolutely need to know about. The first one is the most common one. It's the one that most people are familiar with. The knee jerk, the patellar reflex. When tapping the patellar tendon stretches that, which causes a stretch in the rectus femoris tendon, stimulates these stretch receptors that causes the muscle to want to contract and kick the leg out. So you've seen that one before. The other one that you definitely are going to want to know about is the Babinski reflex. Let me show you. Let me just go ahead to this part here. Not this one. Keep going, keep going, keep going, keep going, keep going, keep going, keep going. There it is. Okay. So here is what the Babinski reflex is going to look like. You've probably seen this in a TV show or a movie. Somebody is getting wheeled into the emergency department, maybe from a car accident or something. And while they're getting wheeled in, there's going to be a doctor or a nurse or somebody's going to grab the patient's foot, and then they're going to get the opposite end of a reflex hammer, like this reflex hammer here, but they're going to use the end that you usually hold with, and they're going to stroke the person's foot. And they're going to stroke the lateral side of the person's foot from the heel up and around to the toes. So a lot of people automatically think, oh, they're looking to see if the person has sensation, but that's not really it. When we do this and we stroke up the foot from the heel all the way up and around the toes, like you see here, we're going to watch to see what the toes do. Because what we want to see is we want to see the toes bend downwards. We want to see them flex the toes. That is normal. We call that a negative Babinski. Negative Babinski is normal. That's good. If, when we do this, the toes dorsiflex, go the other direction, and fan up and away, that's positive Babinski. That's abnormal. That could indicate something like a spinal cord injury. The toes fanning up and out. So curling down is good. That's normal. That's a negative Babinski. Fanning up and out is bad. That's positive. That's abnormal. Positive Babinski is abnormal. This is true in people except for children, especially those young children, because in young children, it's the opposite. Normal will have their feet fanning out, their toes fanning out. Abnormal will have their feet curling down. And that's true up to about 18 months when they're walking. So the Babinski is one that I think you definitely need to know about. Oh, and let's go back to this picture. Here we are. You can see here, when you want to test this, find the patella, the kneecap, and then go just beneath it. You can see where the patellar ligament is. And you can feel, your kneecap, and you can feel just below it. And you give a little tap right there. And that is going to cause the stretching of the quadriceps femoris tendon as well, not the ligament, the tendon as well, which activates those stretch receptors in the muscle, which send the signal up to the spinal cord, which interacts with that interneuron, and then it sends a signal down that motor neuron to tell that muscle to contract. So you see, when we're doing this, primarily we're testing the spinal cord and the nerves to and from. Although other things can also alter reflexes, but that's what the test is specifically done for. Okay, so those are the two that I want you to know about, patellar reflex and Babinski. Autonomic does things automatically. And I said that most of our lives, we are living under parasympathetic division. What I call rest and digest. Only when we're being chased by a bear do we switch into sympathetic division, fight or flight. Time to put up a fight or run away. When it comes to bears, I prefer to run away. Now, obviously, it's not just bears, but going through that haunted house that you paid $18 of your hard-earned money to walk through just to get scared. Or maybe you're driving home and you're in a hurry because you have to use the restroom, so you don't make a complete stop at that stop sign. And then you see those police lights go on behind you and you're thinking, oh, and I didn't pay my insurance this month. Your body switches over into this panic mode. That's the sympathetic division. But I always say being chased by a bear, because I think most people understand what that means. I mean, not that you've actually been chased by a bear, hopefully. There's one thing to always remember, however, when you're being chased by a bear. Always remember that you do not have to run faster than the bear. You just have to run faster than the person next to you. I guess that goes with zombies as well. Okay, I think that is. We talked about some of that already. See, this right here starts to get a little too detailed. In the green side, this is like the parasympathetic pathways, and in the, like, I don't know, peach colored side, these are the sympathetic pathways of how messages are getting sent to these areas. So yes, it's good stuff to know, but I think this might just be a level where it is a little too much. Knowing that they exist is perfect for right now. You can learn those. Oops, come on. You can learn those another time. All right, so I'm not going to talk about that. We saw that. This is an interesting fun fact. At least 75% of all parasympathetic preganglionic fibers travel in the vagus nerve before synapsing with the postganglionic fibers somewhere in the chest and abdomen. Oh, that was interesting. That's the only one, sure. Another thing that I would, maybe not so much in this class, but good stuff to know about. Axon terminals of autonomic neurons, remember these are things that do things automatically, use either norepinephrine or acetylcholine. Now you'll remember we talked about acetylcholine with muscles. Remember, the signal comes down as an electrical signal, which then converts into a chemical signal called acetylcholine that goes across the neuromuscular junction, binds to the receptors on the muscle, which then causes the sodium to come in, et cetera, et cetera. So that one we've heard about. Norepinephrine, we have not, but you may have heard of epinephrine. Epinephrine is also known as adrenaline. That is a hormone. Norepinephrine is a neurotransmitter. There is a slight difference between them. Epinephrine has an additional methyl group. I think we talked about the methyl groups, the CH3 groups, back in chemistry, maybe just briefly. But there is a difference between the two of them, norepinephrine and epinephrine. So the adrenergic fibers have the axons that release norepinephrine. The cholinergic fibers have the axons that release acetylcholine. So you can see that talking about neurotransmitters and things like acetylcholine, for instance, if you don't understand the very basic of a nerve releasing that neurotransmitter across that space, when things start to get more complicated, like the adrenergic and cholinergic fibers, it's going to be really difficult to understand it if you don't understand this earlier stuff. So that's why I'm trying to not spend a whole lot of time on this and spend a whole lot more time just explaining how acetylcholine crosses that neuromuscular junction. It is that chemical message. Norepinephrine is that chemical message. Remember, we're going to have an electrical signal that becomes a chemical signal that becomes an electrical signal. The chemical signal, or the chemical message, as I call it, norepinephrine, acetylcholine. Let's just look at this basic junction again. Let's not worry about what type of receptors there are. But remember, the signal comes down this axon right here in the form of an electrical current. When it gets to the very end, that electrical current, that electrical message has to be changed into a chemical message. So now we have a chemical message that is released. In this case, I think they're using acetylcholine. Yes, acetylcholine, which then crosses that space, that synaptic cleft, and binds to receptors on the next neuron. That's going to cause sodium to come rushing in, more positives on the inside of the membrane of this cell. So now, because we have more positives on the inside of this cell, that's changing back into an electrical message again, which is eventually going to get sent down here and released as a chemical message. In this case, they're showing norepinephrine, which crosses that space, and et cetera, et cetera. What was on here? I think, now I'm not going to get into monoamine oxidase inhibitors. I think that is just about it. Major function is sympathetic division, fight or flight. Sympathetic, fight or flight. Parasympathetic, rest and digest. Parasympathetic, rest and digest. Yeah, that's it. Wow. I am surprised. I finished out exactly, or almost exactly 12 o'clock. Okay. Good. So, like I said, there were some areas I needed to kind of zip through a little bit. Going into any more detail would have just been wrong. There's no reason to confuse you. I know this stuff is difficult as it is. You've heard people compare things that are difficult, but then they'll say, it might be hard, but it's not brain surgery, meaning brain surgery must be really, really complicated. Yeah, because the nervous system is really, really complicated. And as you can see, as we get down into the levels of the different neurotransmitters and things going across, it becomes more and more complicated. So just a brief overview of that is all you need. But you do need to hear about cranial nerves. You do need to hear about the different reflexes, especially the patellar and the Babinski. If I had to choose two, which I did, those are always the two that I would choose. You've got to know parasympathetic, rest and digest, sympathetic, fight or flight. So some of those things, yeah, I definitely wanted to pull out of there. Okay, so now, do you have any questions? Not about the material we went over. Don't ask about the test. Don't ask me my feelings on, the last Jurassic Park movie. About the stuff that we just covered. Any questions over that? It was a lot, right? A lot to see, if nothing else, a lot of visual stuff going on. Okay. I'm just going to check something real fast. Because I know exactly how many times the midterm review had been viewed before I started this morning. As we put more and more protons in it, was viewed 177 times. So since 9 o'clock this morning, it's been viewed 12 more times. That's a lot.
That's because we want to be prepared.
You should be listening to the lecture.
It might not be us. It might be the other class.
They already had the test. First of all, you just said we want to be prepared. We want to be prepared.
They're allowed to check in.
All right. Well,
We
I'm
just
going
like to hear your voice.
Oh, there it is. Okay. Well, at
Dr.
1
Sturgeon,
o'clock.
are you gonna have this? Are you gonna have this on YouTube also? We have a video for this lecture.
The one we just finished
Will
now.
there be a video?
Yeah. Hold on.
Yes.
In fact, I'm gonna end the recording right now. So I'm gonna turn the recording off. So this will effectively end the lecture. Since somebody else had any other questions about that. So let me end